All right, in this presentation, we want to consider post-transcriptional control of gene expression. So again, as I mentioned before, most of the time when people talk about a gene being expressed, they usually refer to just the transcription. And so they'll look for mRNA abundance, and they'll say, oh, this gene's being turned on, or they'll look for a reduction of a particular mRNA species and say, oh, this gene's being turned down, suggesting that transcription is going up or down. And that isn't always necessarily the case. So we've looked at means to control transcription, but we do want to consider then all the other things downstream of transcription that can affect the ultimate outcome of the information that is being converted into a protein to do something. So we want to think about RNA processing. Alternative splicing is always a point of control. mRNA degradation, how long does that mRNA molecule hang around for? The longer it is, the potentially more protein it can produce or can be produced from it. Translational initiation. You may have mRNA, but if the ribosomes aren't allowed to bind to it to translate it, you aren't going to get a protein. And so that's another point of control. And then finally, once I even have the protein, that protein may not be active until I process it, modify it, and then once it's active, how long is it around for? If I break it down immediately, it will only have a burst of activity and then go away. So these are all levels of control, again, creating a very, very complex system that can be very, very fine-tunedly adjusted. And that ultimately is what we're after. So again, just to start to review those, we have alternative splicing, right? So I may be getting different proteins from the same mRNA transcript. So unless I'm looking at that spliced product, that final mRNA, I may be seeing information that is not true. Okay, so a gene may be turned on, I may get a primary transcript, but then which mRNA is actually being produced can differ. How long an mRNA is around is also going to have a huge impact on how much protein can be synthesized from it. So some of the impacts on the life of an mRNA is its poly-A tail, the number of RNases that are targeting it for degradation, lots of control points there. Now, again, once I have an mRNA, I actually have to translate it. So I can block the translation of a particular mRNA with certain regulatory proteins. This could be to give me a kind of a jump start. It's like I'm ready, I want to have this protein product, but not quite yet. But I'm only one step away from it. So I have plenty of mRNAs in the system ready to roll as soon as I get the signal. Or you may want to coordinate the translation and hence the appearance of certain proteins from different genes that are being expressed at different times. So all levels of control are going to ultimately give the cell more flexibility. Now, finally, protein processing and degradation. Again, once I have a protein translated, it may or may not be functional. And there may be things that I have to add to it or take away from it to make it work. So that's a whole nother level of control. In addition, once I have an active protein, it could actually be degraded. Now, the degradation process of proteins in cells is very complex and it's manifold. There are many ways to do it, but one of the ways that's really, really important it's emerging in more and more roles in the cell is the proteasome. And the proteasome interacts with what's called ubiquitin. And so if you Google ubiquitin, you'll just get a tremendous number of hits. There's lots and lots of information out there on ubiquitin and ubiquitination of proteins to target them to this large barrel-like protein structure, the proteasome, that's literally going to chop it up into small peptides. So again, things in the cell that we're discovering all the time that lend themselves to more control of the biochemistry that's going on. Now, this is just touching on so many topics that are so deep. There's so much going on here, but I just want you to have heard some of this stuff, and you can read more about it. But really, when we look at the genome of most eukaryotic organisms, what we find is that a very, very small percentage of it actually encodes for protein. And so for the longest time, people, which I never use the term, and I hate it, is when people use the term junk DNA. But there was all this extra DNA that didn't seem to do anything at all. It's just junk DNA. Well, what we're learning now, that DNA that doesn't appear to encode for proteins may in fact code for functional RNAs. So non-coding RNAs that, as we'll see, and we've talked about this idea that RNAs can be catalytic as well. They can fold back on themselves, they can bind to things, they can form a shape. They're going to be doing lots of stuff. So that junk DNA, we're finding more and more, may be controlling things like what we call micro-mRNAs and small interfering RNAs. And this is an incredibly powerful understanding of how the cell works. And so when you look at microRNA transcript, there's a whole process to make these smaller microRNAs that are going to interfere with mRNAs that encode for protein to target them for degradation. You're going to talk about small interfering RNAs. This has been built into a whole technology. They can interfere with the regulation of access to the DNA that we talked about with chromosome at a chromosome level. But most importantly, and this is really the thing to take forward into your career, is this idea that now I can actually engineer what's called RNAi technology, interfering technology. So these small interfering RNAs, I can produce them artificially, inject them into a cell, or create a gene in a cell that I have control of turning on or off. And by doing that, I can then control the functionality of any gene that I have a sequence for. And basically, I have the sequence for all the genes because I can sequence your entire genome. So whereas before we had to spend a lot of time randomly mutagenizing a creature to try to get a knockout or a dysfunction of a gene of interest, now if I have the sequence, I can just turn it off. And it is beyond expression to how powerful that technique is and how hard it was to accomplish that before. So you're heading into an era where this stuff is going to be every day for you, but when I first started in research, it was a huge deal to do this. So I want you to really be familiar with this idea of RNAi technology.